I'm Dr. Aman Chugh. I am the chief editor at afibprofessional.org, an HRS ACC community on Cardiosource. And I'm here today with Dr. Sanjeev Narayan to discuss the results of the PRECISE trial. Dr. Narayan is a professor of medicine at the University of California at San Diego and the VA Medical Center in San Diego. Dr. Narayan, thank you for joining us today.
Thank you very much.
And would you tell us a little bit about what you looked at in this study and what you found?
Yes, certainly. So the PRECISE trial is a bit of a mouthful. It stands for the Precise Rotary Elimination without concomitant pulmonary vein isolation for the successful elimination of paroxysmal atrial fibrillation. The basic hypothesis was that the elimination of so-called rotors or focal sources for atrial fibrillation would result in the successful elimination of AFib in patients with paroxysmal AFib without performing pulmonary vein isolation. The study was motivated by a series of studies, including work from several investigators over the years showing that AF can terminate before pulmonary veins are isolated, that pulmonary veins, though triggers, may not be sustainers, and also by our study a few years ago, the CONFIRM trial, that showed that when you took patients who had a variety of AFib types and targeted these rotors or focal sources and then did PVI, the success rate was far higher when you did both versus just PVI. So CONFIRM didn't answer two questions. One was, how would this work in paroxysmal AFib alone? And second of all, how would it work without pulmonary vein isolation? And that was the premise for the study. So this was a 5-center study, single arm, that was designed to test that hypothesis. We enrolled 31 consecutive patients. They were standard paroxysmal AFib patients. They had standard indications for ablation. They had never had AF ablation before, and there were no exclusions per se, except of course they consented to the protocol. We brought them to the lab. Once we had atrial fibrillation, then we performed mapping in a fashion that we've recently described, so-called firm mapping, where you place basket catheters into the right atrium, identify rotors or sources, burn them, get rid of them on remapping, do that in the left atrium. Then we would check the pulmonary veins. We wouldn't isolate them, but we would check them, and then we would stop. All patients got that protocol. Then we followed patients for long-term outcome. The acute outcome of the study was elimination of sources, and the long-term outcome was elimination of atrial fibrillation, measured in the standard fashion, which was if patients had an external monitor, 30 seconds was a failure. If they had a continuous implanted device, then less than 1% on that cumulatively. We followed patients quarterly. Drugs were stopped at 3 months, and 65% of patients had an implanted device. The results of the study were that the patients had a standard demographic you would expect, 59 years of age. A few had failure. The CHAD score was 1.5 on average. When we looked at the procedural acute results, we saw that all patients had sources, rotors or focal sources, predominantly rotors, 2.5 each. There were 2/3 in the left atrium, but interestingly, 1/3 in the right atrium, where you wouldn't normally ablate for paroxysmal atrial fibrillation. We were able to terminate and eliminate these rotors. We didn't perform any PVI according to protocol, and the overall ablation times were 17 minutes on average for the AF ablation part, of which about 1/3 was in the right atrium because a 1/3 of the rotors were in the RA. And the total ablation time, including CTI flutter lines, which were needed in 9 patients, was about 22 minutes. So the acute outcome I've described, we managed to eliminate the sources. The chronic outcome was based on the implanted monitors and event monitors in a 1/3 of patients. And the single procedure outcome was 82% freedom from atrial fibrillation by those criteria. Median follow-up 2 to 3 days. And so we concluded from that study that paroxysmal AFib, contrary to what we've previously been thinking, is a substrate-based disease, and that it can be treated effectively by eliminating rotors or focal sources without PVI.
Wow, these are very impressive results and do make us think of the patients with paroxysmal atrial fibrillation differently. So how do you see these data impacting our care of ablation management of patients with paroxysmal atrial fibrillation?
That's a great question, Aman. I think that the way we're doing AF ablation currently clearly works for some patients, no question. And I think that's partly because we eliminate triggers, and partly because I think current lines we perform might be inadvertently targeting rotor sites. I think we've shown that in some other recent work. So it would be nice if we brought all of our patients up to the success rate we see with some of our more successful patients. I think that one step would be to target these sources more effectively in more patients. And I think one approach would be to do pulmonary vein isolation with this so-called firm procedure. Of course, I think based on these data and others that will be forthcoming soon, perhaps firm-only ablation could be performed. I don't think it really matters as long as we eliminate sources. And I think that is the key component I'm taking away from the study. Much like other rhythms, essentially all rhythms we treat, SVTs and even VTs, we take out the substrate. I think the key for AFib, even paroxysmal, is also to take out the substrate.
Gotcha. And what do you see the next steps going forward?
Yeah, that's also great. So the immediate next steps will be to collect more patients, which we have already done. I think that there are other ongoing trials which are being planned by independent investigators to do a blinded comparison, randomized comparisons between firm versus PVI in various populations.
That sounds great. Well, thank you once again for taking the time to share these very exciting data.
Thank you, Aman. Very nice.
Thank you.